So I have a small laboratory in this hospital. We are like 4 people in the lab, and so we work on epigenetics. So this is liver disease, the classical progression of liver disease. So you see there healthy. There's no point, right? Just, I go like that. Okay. There is a
something like pan or something.
No, it's all right. I mean, it's fine. So. The most common liver disease is called steatosis. So it's the accumulation of lipids inside the liver. You can see the histology and this is induced by overfeeding, so Western diet. So if you eat too much fat, so I put there burger from McDonald's, you have an accumulation of the fat inside the liver. This is very common. 1 people out of 3 has this, and normally it's silent, so it's benign, nothing, no symptoms. However, if you have an extra stress, such as inflammation, reactive oxygen species, or lipotoxicity, for instance, you may evolve in another disease state, which is called NASH, non-alcoholic steatohepatitis, characterized by inflammation, and this is a disease. So you have symptoms, you must get treated, because eventually it can evolve into cirrhosis, which is the end-stage liver disease that normally needs transplantation as only therapy or remedy to cure it. And in some cases, the cirrhosis can also evolve into hepatocellular carcinoma, which is a very serious cancer. So normally people that make research on cancer, on any kind of disease, focus on genetics. So polymorphism, what genes are wrong. But as you know, not all our destiny is in the genes, in the DNA. But this actually, the new science that you know very well is epigenetics, that basically this is a cover of the Time magazine a few years ago. That is the science that study how genes are regulated in a way that does not depend on the sequence. So anything else which is not the sequence. So this is a picture of the chromatin. So you see that the chromosome can be more or less compacted, and the functional unit of the chromatin is called the nucleosomes. That is a bunch of histones. There are 8 histones around which the chromatin wraps. So you have this sort of pearls, chain of pearls. And the opening or closing of this structure depends from many mechanisms. There you have RNA polymerase, long non-coding RNA, enzymes which are called the methyltransferase, which add methyl groups to DNA, so it's all very, very complex. But in our lab, we are particularly interested in one mechanism, so we can study everything, so we just focus on one thing. So you see up there, there is the nucleosome. As I mentioned, it's a core of 8 histones, H2A, H2B, H3, H4, which is repeated twice, so that makes 8. And it has been found that these are called traditional or canonical histones. There are other histones called the histone variants, which can take the place of the normal histone and can regulate the gene expression. So one of this guy is called macro H2A. It's a natural occurring variant which has the ability to substitute for histone H2A. So this guy can take the place of histone H2A. And this I'm going to talk about this for most of my talk. It is clear, there are some reference, that this histone, which is large, macro from the Greek means large, it's about 45 kilodaltons. Instead, you know, a histone is usually 15, 17 kilodaltons, can regulate transcription and cell metabolism. So this is a whole field. So we know, we are studying this histone, macro H2A, which is involved traditionally in repression of transcription, but there are others. So you may have heard about H2AX, which is a DNA damage. H2A at Z, H2A BBD. So there's many of these histone bias. So this complicated graph, maybe I can do like this. No. Way to point things out. So this is a technique called ChIP-seq, chromatin immunoprecipitation sequencing. So these graphs, these peaks, are places where the histones bind the DNA. If the histone bind the DNA, this technique produces a peak that means a binding. And basically, you have up on top you have RNA-seq, which is basically the technique that uses RNA polymerase as a protein that binds the DNA. So this is a snapshot on chromosome 1. It could be any chromosome, but we're just looking at chromosome 1. You see in orange there are some peaks produced by RNA polymerase. Then just look at the blue, is a macro H2A1. You will see that where there are the orange peaks, the blue peaks are absent. And where there's no orange peak, the blue peaks are there. So it's since that is the RNA polymerase, which is a transcription, means that it's involved in a repression of transcription. So this protein contains this domain which is called the macro, but this domain is not unique for this protein. It's actually contained in many proteins, and notably also in PARP, poly-ADP ribose polymerase protein. So macro domain is quite spread in nature, and goes not only in mammalians, but for instance, this is a fungus, Archeoglobus, actually no, sorry, this is Archaea, is a bacteria. This fungus, this bacteria, Archaea, has a protein that contains macrodomain. This is a three-dimensional reconstruction of the macrodomain from the Archaea and from human histone, macro H. And you can see that it's a very similar structure. So it's conserved across evolution, so it might be important. Okay, now I show you about, oh yeah, you just missed it, I talked about PARP. So we have mice which are knockout for macro H2A1, and this is an important tool to study the role of the protein, because we can see what happens if you don't have the protein in a mouse. So as it's written here, knockout mice for the protein develop and grow normally. There's nothing wrong with them. If you feed them a CHO diet, there's nothing wrong. It's a non-striking phenotype. However, if some people look at the liver and they saw, they wanted to look where, so it's a histone, so it binds the DNA, so which genes it binds. And they found that the majority of genes are genes that are involved in lipid metabolism. So lipoprotein lipase, lipoprotein receptor, fatty acid transporter, and so on. So preferentially this histone binds genes involved in lipid metabolism. So this is the same burger before. So basically we wondered if we feed the mice high fat diet, so we make the mice fat. What happens if in mice that don't have macro H2A1? So this is a study which we recently submitted. So we find that the mice that are fed a high fat diet and don't have the histone accumulate less body weight. So they have less body weight. So if you look at this, this is called the DEXA scan. So it's a scan that gives in color the fat mass and the lean mass of the mouse. So you have there low L, it's low, high, it's fat mass. So the dark color, purple, means lean mass. The yellow means fat mass. So if you look at this mouse here, compared to that one, there's much more purple. So it's a more lean mass. And this is also, and less fat mass. And this is also represented on the left by the histograms. So these mice also have lower levels of glucose in the blood, so fasted glucose, so they are less glycemic. However, when you inject insulin, there's no difference. We also done this called the OGTT. It means oral glucose tolerance test. So basically, you inject the mice with some, or you give the mice some glucose. It happens also to humans. So you have a peak in glucose concentration, and then the pancreas produces insulin, and the insulin produces a decline in glucose concentration because the body metabolizes the glucose. You can see that in blue, the knockout mice, so mice that don't have the histone, clear out the glucose from the blood much faster than the normal mice. So these are called glucose tolerant, so they are more glucose tolerant. This is a quantification of the graph that I showed you above. However, there's no difference in insulin tolerance test. The same test you can also do with insulin. So the mouse, also you can do in a human as well, has some glucose in the blood. If you inject insulin, the glucose will clear out, but then it will restore. So it's this kind of curve like that. And there's no difference. So basically, they are more glucose tolerant, but there's no difference in insulin sensitivity. So you know that insulin exerts its effects by activating a pathway which relies on ATT, phosphorylation of ATT, and we measure the ATT phosphorylation in the skeletal muscle of the wild-type mice and knockout mice on the high-fat diet when you inject insulin. And you can see that, just look at the graph, because this is many bands, that the knockout mice have an activation of the ATT in response to insulin. It means that they are more insulin sensitive in the skeletal muscle. In the liver, there's the same trend. Also in the liver, they are more insulin sensitive. However, there are no difference in triglycerides and cholesterol in the blood and also in the liver. However, a very interesting thing that we observed at the first slide that they show you, I told you that when you have a fat in the liver, it's a risk factor to develop steatohepatitis, inflammation. Inflammation is induced by infiltration of inflammatory cells in the liver. So what happens is that you have the blood through the portal vein, goes to the liver, and if you have inflammation, there is an attraction of all these inflammatory cells to around the portal vein. And this is called portal inflammation. That's what we see here. So that's the portal vein on the left. So in the wild-type mice on the high-fat diet, you have all these blue cells around the portal vein. And instead, in knockout mice, we don't see many inflammatory infiltrates around the portal vein. So this is also the quantification below. So the mice are protected against inflammation in the liver. So the conclusion of this first part of my presentation is that the causative absence of macroH2A1 in a mouse protects from increasing body weight upon a high-fat diet. In these mice, there is insulin hypersensitivity in the liver and the skeletal muscle, but not in other tissue. And a decrease in hepatic portal inflammation is observed in these mice. So the phenotype is a bit mild, but is convincing to us. And now this paper is a revision in the International Journal of Obesity. So the absence of the histone is beneficial if you have less of this histone. Okay, now I go more complicated. So I hope not to lose you. If it's something not clear, please interrupt me and ask. So this histone, like many other protein, can go can undergo alternative splicing. So the messenger RNA can be spliced, can produce two isoforms, two different proteins, which are called the macro H2A1.1 and macro H2A1.2. This is called the alternative exon inclusion. So you have the structure of the genes up there. The boxes are exon, in the middle are introns. So in one case, one exon is included, and the other case, the other. And just to clarify, the mouse I showed you before doesn't have any of this protein. So the whole gene is depleted. So the difference between this isoform is that small metabolites like ADP ribose that is used by PARP, for instance, can bind with very high affinity macro H2A1.1, but not a macro H2A1.2. So what you're looking at, these are called the calorimetry. It's a very easy test. You take an Eppendorf water, you put your protein, you put ADP ribose, you put into a machine which measure the heat. So if the protein interacts, energy is going to be released, so you have a heat. So you have there time, across time, so there is a heat released across time. So this is a macro H2A1.1. Macro H2A1.2 does not bind metabolites. So this is the difference, the functional difference, the only one we know. And this is, was published in 2005 by my ex-laboratory. These metabolites, ADP ribose, not only they're produced by PARP, but they're also produced by sirtuins. I don't know if you heard about this enzyme. There's a lot of interest because they're activated by resveratrol. I know some of you work on this, and also by polyphenols that they contain in red wine. So the enzyme, the sirtuin, produces this metabolite, and the metabolite binds the histone, but only one of them on the left there. Instead, this one here, macro H2A1.2, does not bind the histone. So, going a little bit into epigenetics, which is a whole science. So you have basically the DNA, you have the histones in the chromatin. The histones can be modified. So these are called histone post-translational modification. So you have like acetylation, methylation, and a whole science developed in the last 20 years. Where the people identified the protein domain that bind the histone modification. So you see there acetylation is recognized by bromodomain, methylation is recognized by chromodomain, and the histone can be also ADP ribosylated. And this is a new, this is in technical jargon, this is called readers, chromatin readers. So the ADP ribosylation can be read by macrodomain. So it's a new marker that we propose. So now I go a little bit more functional. So what is known about this isoform? So it's known that the isoform that binds the metabolite anti-correlates with cancer aggressiveness. So in the upper part of the slides you have immunohistochemistry of lung cancer. It's too bad really I don't have a pointer, but on the left you have a Ki67, which is a marker of proliferation, and you see let's see if I can here. So you see here, this is a negative for Ki67, which is a marker of proliferation. This is very positive for 1.1. Vice versa, where Ki67 is positive, 1.1 is negative. Same for, this is colon cancer. So Judith Sporn, which is a colleague, she took cases of colon cancer, human colon cancer, and she clustered the immunohistochemistry for the histone, macro H2A1, on the base of the expression. So she took 3 groups: strong expression, strong expression, intermediate expression, and also, so this is strong, this is intermediate, this is weak. And you can see that this is the survival of the patients. So patients who have a weak expression of macro H2A1, they die, most of them, in time. So this is months, so in 5 years they're all dead. Instead, the patients which have a strong expression of the histone, almost all of them survive. Intermediate expression, intermediate survival. So, of course, these are small numbers, like 20 patients, but still it's significant. Also in cells, this paper, just look at the line with the dark boxes. So these are 2 cell line, A459, and the HeLa cells. If you overexpress the histone, macro H2A 1.1, so again look at the line with the black boxes, you have a decrease in proliferation. So it's anti-proliferative, not only in immunohistochemistry but also functionally. So as I said, my main focus is the liver. So why I told you all this? What is the role of this histone in hepatic metabolism and disease? So this is just in immunohistochemistry that shows you that in the liver of a mouse, both isoforms are expressed. So this is immunofluorescence, and you can see the nuclei. DAPI is basically the nuclei staining. On the left, the green is the histone, and the merge shows you that you have a good colocalization. So, again, I've shown you this progression of liver diseases, so steatosis, NASH, cirrhosis. That's in human, but how we can model it in animals. So there are several models, but here we used 2 models. One is called the high-fat den. So what is this? This is, so if you take a mouse and you give a high-fat diet, the mouse will never get cancer, never ever. So, but if you inject the mouse with a little carcinogen, which is called DEN, diethylnitrosamine, it will develop cancer. It depends on the dose. If you inject a small dose, basically the mouse will not develop cancer. But if you treat the mouse with a high-fat diet and inject a small dose of the carcinogen, the mouse will get cancer. And so that's what you see here. This is a mouse with a high fat in the liver, and triglycerides in the liver, and then also has cancer. And this is a dietary model. We have also a genetic model. So genetic model is the P10 knockout. So what is P10? P10 is that's a scheme over there. So insulin through PI3 kinase phosphatidyl inositol 3,4,5 phosphate activates AKT, which can induce steatosis and cancer. P10 is a protein that blocks the production, the activation of AKT. So if you don't have this protein, this is a liver-specific P10 knockout. That's what the liver looks like when the mouse get aged. It's disgusting. So the liver, they die at 8 months. So the liver gets full of fat and also full of cancer. So we took these 2 models to look at our favorite histones. And we found, if you look at the red boxes over there, that in a condition that develop cancer, so high fat plus DEN, or old mice which are knocked out for P10, there is an increased expression of the histones. So they are also marker of cancer, liver cancer. But it's different, because what I showed you before was that one histone was associated to protection and the other not. Instead, both of these histones are associated to disease in liver cancer. And the proof...
H3.
You have shown the H3 or not?
Yes, H3 was a marker of...
it was marker of loading.
Yes.
Not
Not acetyl, no, total.
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 It's the marker for the loading.
Yes, exactly. With the market for loading, for Western blot. But we're even more convinced when we look at the humans. So these are human biopsies. You have on the left the steatosis, and on the right you have the cancer or the area near cancer, and you see also the quantification. So pretty much all nuclei in cancer are positive for the histones, whereas in steatosis, no, this is not the case. And even these nuclei that you see here, these dots here, there are black dots, but these are not hepatocytes. These are they are in the sinusoidal spaces, so they can be like Kupfer cells, hepatic cell cells, like other cells, not hepatocytes. So and this we published last year in PLOS One. So then, and that's what we're doing now. Just came out with a paper on this. This is our observation. So we see the isoforms are increasing cancer. But so what do they do really at the functional level? So we took cells, EPA16 and EPG2 cells. These are hepatoma cell lines that now Nina has to try to culture with more or less success. And we transfected with the proteins, the histone, which are coupled to cherry protein. Cherry is the fruit, but it's like is like RFP, red fluorescent protein. So if you transfect the cells, they become red, and these are orange because it's a merge with the double staining. So it's red plus blue becomes orange. So when we have these cells like this, we measured the accumulation of lipids and cholesterol, so triglycerides and cholesterol when we treat the cells with high-fat diet. So high-fat diet with free fatty acids. So you put on the cells free fatty acids. Then you measure triglycerides and cholesterol with the commercial kits. For example, Roche sells some of them. And you can see that the middle column, always the middle column, where the macro H2 1.1 is overexpressed, basically they're protected from the accumulation of triglycerides and also cholesterol. So in the upper panel is, in the left panel is EPA16, and this is FG2, but it's always the same result. So 1.1 protects. And then we did something quite fancy, which is the gene expression. So this was the experimental workflow. So basically we took the cells, show you, transfected with cherry. We treated with free fatty acids. We extracted RNA, and then we did arrays. Not microarrays, but we did a bunch of genes, 84 genes. We measure 84 genes, which are involved in fatty liver. So QIAGEN sells, I don't know if you're aware of this, it sells PCR assays of your, about the pathway that you're studying. So if you're studying inflammation, they sell a plate on 85 inflammatory genes, or it depends what you're interested in. And then we did some system biology. So this is a complicated slide, but so when you have metabolism of fat, you have specific processes going into the cell. The fat can be metabolized. That's called the beta oxidation. The fat can be transformed or can be the result of transformation for carbohydrates. And that's carbohydrate metabolism. Cholesterol can be metabolized or transported, and that's the third one. Or there's many others genes involved in lipid metabolism. So this is difficult to read, but in each of these four processes, four clusters, the first column it's cells which overexpress 1.1. In the second column is cells that overexpress macro H2 1.2. The colors is indication of expression. So you see they are completely different. So, but this is not functional. This is just for the eye to visualize. It's something that you put in the software. It gives you, oh wow, there's a difference. But what we did, we had the help of a bioinformatician. So this is you have to look at this like that. Each ball is a gene involved in lipid metabolism. The balls are connected functionally, and so you have the blue traits. The thicker is the trait, the stronger is the functional interaction. And and then aside each gene